Gel electrophoresis is a molecular biology technique used to separate DNA fragments based on size. In this demonstration, we will prepare an agarose gel, load DNA samples and ladder, run the gel, and visualize the DNA bands using an e-box. To prepare the gel, we first weigh 1.5 grams of low electroendosmosis agarose and dissolve it at 100 millimeters of 1x TAE buffer. This produces a 1.5% agarose gel, which is ideal for separating medium-sized DNA fragments. The solution is heated in the microwave until the agarose is fully dissolved and clear. Heating melts the agarose so it can form a uniform gel matrix when cooled. Once cooled to about 55 degrees Celsius, 5 microliters of DNA run-safe stain is added. This allows DNA to be visualized later under ultraviolet light. The agarose is poured into the casting tray with the comb in place to form wells for the sample loading. The gel is left to solidify until firm and opaque.
Next, the DNA ladder and molecular weight ladder are prepared in labeled microcentrifuge tubes. To each PCR sample, we add DNA run-safe loading dye and glycerol. The loading dye helps track DNA migration, while glycerol increases sample density so it sinks into the wells. The DNA ladder is prepared the same way and serves as the molecular size marker for estimating fragment length. Once solidified, the gel is placed into the electrophoresis chamber and submerged in 1x buffer until fully covered. The DNA ladder is loaded into the first well using a steady hand and fresh pipette tip. Each DNA sample is then carefully loaded into separate wells. Proper loading is essential to avoid puncturing the wells or cross-contaminating samples. The chamber is closed and connected to the power supply. DNA is negatively charged because of its phosphate backbone, so it migrates toward the positive electrode. When voltage is applied, smaller DNA fragments move faster through the agarose matrix, while larger fragments move more slowly. This separates DNA by size.
After electrophoresis, the gel is transferred to the e-box for bands fluoresce under ultraviolet light, revealing separated fragments. The ladder is used as a reference to estimate the size of each. Gel electrophoresis separates DNA fragments by size using an electric field and agarose matrix. It is a simple but essential technique for DNA